Hello, my name is Monica Chu and I will be discussing the experiment Oxidative stress by Juglone causes death in GFP-Htt-PolyQ C. elegans over time. Huntington's disease is a progressive neurodegenerative disease caused by extended CAG repeats in exon 1 of the HTT gene, which translates to the HTT protein containing an extended PolyQ tract. When PolyQ repeats reach 40 residues, this results in aggregation of HTT in the neurons and an onset of Huntington's disease. To study Huntington's disease in a biological laboratory, C. elegans is a valuable model organism. Its significant utility in Huntington's research is due to the establishment of its cell lineage, which includes all the neurons of the adult hermaphrodite, and that they are easy to observe and manipulate in a laboratory. Transgenic C. elegans were created in a previous study by Wang et al. In 2006, investigating the role of ubiquitinin on regulating Huntington's development. Worm lines created in this study can be seen in this figure, which is figure 6 found in the study's article. In part B, GFP fusion to HTT allows visualization of HTT aggregation. Worms with 74 glutamine repeats show aggregation of HTT as fluorescence of the protein is seen in clumps and spots along the body of the worm, while worms with 28 repeats show little aggregation as fluorescence of the protein is linear and spread out along the worm. Another previous study by Hartwig et al. In 2009 used Juglone as an oxidative stressor to transgenic C. elegans by giving it to them orally. They then evaluated how the worms adapted to Juglone over time and if they could still live a normal lifespan. These discoveries led us to become interested in the effect of reactive oxidative species on C. elegans containing different lengths of PolyQ repeats. So we performed an experiment using worm lines expressing GFP-Htt with PolyQ repeats of 28 and 74 and wild-type worms. We induced oxidation to the worms using low concentrations of Juglone solution, 30 micromolar and 60 micromolar, in addition to 0 micromolar, which was just M9 buffer. 3 replicates were performed, and after 30 and 60 minutes of exposure to Juglone, we recorded the number of live worms based on their movement. This led us to these results of percent survival of the worms after 30 and 60 minutes. In these graphs, after 30 minutes, Q74 had the lowest percent survival at 0 and 30 micromolar Juglone, while Q28 had the lowest survival at 60 micromolar. After 60 minutes, Q74 had the lowest survival in all concentrations of Juglone. Also, wild-type worms had the highest percent survival in all concentrations and at both time points. In general, the overall trend was decrease in percent survival over time and lower percent survival in higher concentrations of Juglone. From these results, we concluded that a longer PolyQ length led to lower percent survival when exposed to ROS, and that higher concentrations of ROS led to decreased survival of all the worm lines, with significant decrease in Q74 worms. This suggests that longer PolyQ repeats has an effect on C. elegans survival in the presence of ROS. This effect could mean that HTT aggregation caused by PolyQ repeats impairs the antioxidant defense system of C. elegans, leading to their death, or that ROS quickens the rate of aggregation of HTT, also leading to their death. Further studies of Huntington's disease and ROS include monitoring the fluorescence of GFP-Htt-PolyQ aggregation over time in the presence of Juglone. For this study, I would like to thank Abraham, Nicole, and Anna for their help in the lab, to Biology 18A staff for preparation of materials used, and to Brandeis University for use of facilities.